Our next talk is going to be by Adway. He is a software engineer at the University of Maryland. He is also going to speak to us about work that was recently presented, I believe, at a CVPR workshop on data curation and augmentation in the domain of medical imaging. And then this work was so well received, they awarded it Best Poster. So congratulations. And yeah, we were just sort of, when we saw this again would be a wonderful piece of work to share with the community. People can learn from it, maybe get their own ideas about what they possibly could achieve. And so congratulations, Adaway. And it's been a pleasure seeing your progress over the last few months and so on. Yeah, over to you. We're really happy to dive in and see what's been going on.
Wow, thanks so much. Thanks so much for the introduction, Nick. I really do appreciate it. Um, yeah, it's been my pleasure and privilege to be presenting our work today. And so first of all, can you see my screen? Is everything okay?
Yes, looks great.
Okay, awesome. First of all, again, thank you for this opportunity and for inviting me to talk today at the Flower Monthly. This is really a great moment because I follow Flower Monthly, and it's really great to be uh, sharing our work here today. And so, just a quick intro about me. My name is Adway Kanhere. I am a software engineer at the University of Maryland School of Medicine. I develop algorithms for particular clinical translational applications in mostly radiology. And today I'll be presenting one of the papers that I presented at CVPR on privacy preserving 3D medical image segmentation. And also I'll be talking about our participation in the Flower Next pilot program and the progress that we've made, and just sharing this with the community, and hope that more folks can come in and help develop using Flower amazing techniques for different applications. And so the agenda for today, what I'll be going through is I'll start off with the background and current challenges in the medical domain. And then we'll look at FL as a unique solution, why we can use federated learning in this particular setting, and then some challenges in applying FL in medicine. And then I'll talk about our work, which is Segways, which will be presented as a paper and as a poster. And I'll also be talking about our work with Flower and how we developed Segways and moved that from a research to a production ready system. And then we'll talk about future scopes. And we also have a short demo. It's not a live demo, but we have a short demo. So I'll also go over how anyone can sort of implement the pipeline that we've developed. All right, so to start off, let's talk about, in general, the two big pillars of deep learning, which is data and models. And so this is, again, very crucial in the medical setting, where data curation and availability is a big concern. So I'll be talking specifically today about medical image segmentation, because that is the most common clinical application in most cases, in most medical applications, has several downstream cases as well. The problem with data curation and sort of using data in a medical domain is that sourcing training data to even develop these algorithms is hard, even though medical data is plentiful and easily abundant. And so we have these sort of three V's that we're sort of dealing with in terms of data curation and availability. The first one is volume. So we have large amounts of data, but that needs to be manually annotated. And creating these annotations is expensive and time consuming, because radiologists have to sit down, or clinicians have to sit down and manually annotate these images. And that takes a lot of time. The second concern is variety. So imaging data is non-IID and heterogeneous. And so what that means is, imaging data coming in is usually at a particular site and using a particular scanner or a particular protocol, and that may be different from site to sites or hospitals to hospitals. And so the other concern is veracity. What that means is images have different views. So some scans are full body scans, so they will have the entire body scanned at one go, or some scans can be specific to a particular organ of interest. So you could have a brain scan or a scan of the abdomen. And the last concern is if you have data already annotated, manually segmented, clearing that data is also a concern because it has, there's a lot of legal and privacy concerns involved, especially around sharing sensitive medical data between institutions. And so this is one of the main concerns of the data. In terms of algorithm development, because our data is so scarce and usually annotated for a particular use case, it's usually annotated with a particular organ of interest in mind. And what that leads to is it leads to models that have a narrow focus. And what that means is it will only be annotated for one particular organ, and it seldom can be used for something else. And what this leads to is that you end up training several models which are trained on the same data, but they're trained for different tasks. And this just decreases the clinical translatability and usability of the data and the models that we're dealing with. So the next obvious question is, do we have a solution that can collectively aggregate knowledge from different tasks into a single unified model that you can use to perform all the tasks together? And that brings me to the next chapter, which is FL as a unique solution. And so the answer is yes. Federated learning is the perfect solution for this problem. And so the idea is that you don't want to share the models instead of sharing. So you don't want to share the data. Instead of sharing the data, you would rather share the models. And this can be a really powerful tool because not only will this allow collaboration between different hospitals and research institutions, but this will also allow companies to come in, work with downstream applications without the need to share the data. And so I'll be talking about some challenges in applying FL in medicine. And so this is great, but like let's look at some particular concerns about why this is hard in the medical domain. This is not a straightforward or trivial solution. And the problem with that is because of the problem of incomplete annotations. And so, like I mentioned before, usually when a clinician will come in, they will manually annotate only a particular organ of interest in the data. And so what that means is you will have sites with like different sites, each containing probably the same field of view, so the same organ of interest. So the same field of view, but different organs of interest, and only one organ will be annotated. And so when you're doing federated learning, this can be sort of explained as a client heterogeneity because clients are working with similar data in the same field of view, but they don't have annotations for the other organs typically that you would see in a centralized setup. And some FL specific challenges that also sort of is a concern is firstly, we have distribution shifts which degrade performance. So in traditional federated learning setups, we've seen poor generalizability when you have client heterogeneity, which means clients usually have completely different tasks that they're training on. And we had a paper from Zhao et al., which showed that the difference in data distribution can have such an adverse effect that it can lead to a drop in accuracy by up to 55%. This is for the global model when we're training the global model of the server. The other concern is, again, the global model itself fails to converge in most cases when the data is non-IID. And it's really hard to get a single global model that can effectively aggregate knowledge and have knowledge of different tasks. And in most cases, what we've seen is most global models, they will have good performance on tasks from a particular client, but might fail or perform poorly on tasks from other clients. This is also something that we've seen when we were doing our research. And I'll talk a little bit more about in the next couple of slides. And then the last concern that we've noticed is, the server requires the knowledge of all the tasks before training begins. So there's no way for new clients to come in and join the federation with completely new tasks and new data. And this sort of limits, again, the real world usability or translatability of even FL. We wanted to allow for effective collaboration with clients. And the other concern is that clients, when you have clients with completely different data, you need to have an optimum model training strategy to be able to ensure that the global model doesn't overfit or individual local models don't overfit or underfit during training. And so I'll be talking a little bit about our solution. So what we've seen, and so our motivation for the paper was we noticed that there has been a lot of research around federated learning with non-IID data. Particularly, we've seen a couple of techniques like we have FedBN, which does local batch normalization, and that has shown to be effective in creating in basically combating this problem of data heterogeneity. But we've noticed that it doesn't effective. It leads to more personalized approaches and doesn't lead to a global model which can be trained effectively. And so there was a seminal paper, which was in ICML 2020, which talked about the convergence of Fed average on non-IID data. And the TLDR from that paper was, if you want Fed average to converge on non-IID data, you need to have some sort of dynamic weight decay. And so we were interested in this technique, and so we went to investigate on that. And so what we ended up doing was we ended up collecting data which is openly available, which is open source data from the medical segmentation decathlon challenge, and we set up this data at 4 different clients. And so we looked at, for example, the liver, spleen, kidney, and pancreas data set, and we chose the modality as the CT or computer tomography images. And we had a completely different test set, which was the beyond the training world BTCV data set, which we chose as an external out of distribution data set to test our model on. And so basically from this table, what you want to look at is notice how every data set at the client has a different shape and different spacing, and also a different sample size. And so this was again a non-trivial problem to actually ensure that federated learning would be able to effectively aggregate this knowledge because of just how heterogeneous this data was. And so what we did was we developed a solution called Segways, which works with heterogeneous data. And some of the techniques that we implemented based on prior research was we had selective weight aggregation using a cosine annealing decay function. And so we also implemented selective weight aggregation. And what that means is instead of Fed averaging, which will ideally aggregate all the weights of the model, we only aggregated particular weights of the model, will be called as a representation block weights. And so we split up our usual backbone, which was a 3D UNet backbone, which we use for segmentation, and we only aggregated, say, all the layers except the last 2 layers, which we deemed as a representation block. And the last 2 layers, which were the task-specific blocks, were allowed to train to allow for local model convergence. And so this is an example of a setup that we did. Effectively, we had 4 different clients. Each client will have an individual data set that they're working on, that they're training locally, and then model updates are sent to a global model at the server location. And we implemented the selective weight aggregation and cosine decay. And because of the setup, our setup actually allows new clients to join the federation without the need to even have task-specific knowledge beforehand. Because as training is happening, you could have new clients join the federation and have new tasks that are happening that are being trained on. And because of the way that we designed the model, the UNet backbone, which is only the representation layers are being aggregated, you can change effectively the task-specific layers of the global model, which will allow for new tasks to be trained on as you're going through it. And so the experiments that we ran in the setup, so we ran 4 different experiments. And so as you can see on the top left, the A's, the baseline models. And what that means is we trained individual models, single models for each task. And so this will be the sort of like a baseline approach in case you didn't have FL. The second technique was a centralized approach, wherein we aggregated all the data in one location. And remember, we didn't actually have, we still had incomplete annotations. So it was still like a partial label case because not all of these data sets were labeled for all the different organs. And then the third technique that we used was just conventional, traditional federated averaging, where we aggregated all the weights of the model at the server's end. And then the last approach was Segways, wherein we had selective weight aggregation, which was only aggregating the layers of the representation block. And so what we saw was very interesting. And so what we noticed was from this table, as you can see, segues clearly outperformed all of the other techniques in most cases, in most organs even. And so I'll be going in detail about that a little bit. But from this, you can see one thing to note is that notice how in central aggregation, the central aggregation model actually fails for some organs. And so I'll just be talking a little bit about that too. And so when we look a bit more in detail about the results, what we noticed was with lesser nodes, segues clearly outperforms or performs at par with conventional FL. And as we increase the number of nodes, segues outperforms all other setups, effectively demonstrating effective knowledge transfer or knowledge aggregation. And so this is actually really powerful, because what this means is because as you increase the number of nodes or the number of clients, you're effectively allowing for just the selective weight aggregation is allowing for a stronger knowledge transfer, which is leading to better results. And again, central aggregation fails in the setup because your data is so heterogeneous that the partial labels degrade the performance significantly. So when you look at this at an organ level, again, segues outperforms all of the approaches for all organs except the pancreas. And as you can see, we notice it doesn't perform as well for pancreas, and that probably can be because the structure is just so small in general, and it's a very complex organ when you're segmenting it, that even when you're training the baseline models for this task, you don't really see a high performance. So going back, as you can see here, compared to all the other organs, in the first row, the single data set models, pancreas performs about okay, not really the best model. Not really a high dice score. And again, this is the dice score comparison, which is basically just the overlap between the masks and the ground truths, which is a common metric that we use in medical image segmentation. And so again, what we noticed was central aggregation again clearly fails because when annotations are missing, the model just failed to segment some particular organ. So in the 2-node setup, it failed to segment liver. In the 3-node setup, it failed to segment liver and pancreas. Spleen. But we also notice here that clearly segues has been consistently performing better than even conventional FL in this case. And when we tested this on completely out of distribution data. And so it's even in that case, segues clearly is performing much better. And so when you look at qualitative results, this is what we see. So notice that in the second in the sample. So I'll talk a little bit about how this is organized. You have your CT image, you have your ground truth, which is what the radiologist will come in and manually annotate, and then you have the results of the different models. And if you look at the first couple of samples, you can see they perform almost really well, but you can see how segues actually perform slightly better than in your traditional FL setup. And when you look at the pancreas, which are in red, what you can see is if you look at the fourth option, the fourth row, you'll notice how segues is the only one that actually approximates a small region of the pancreas. It's because, again, that's such a complex organ that it takes a lot of data to be able to train that really well, and all the techniques just fail. And if you look at the central aggregation, again, you'll notice that central aggregation just straight up fails to segment particular organs in most cases. So this was all great. So now this is the fun part is how can we implement this in a real-world setup? And that's exactly where Flower comes in. And we've been super excited about this, and really it's been great to work with Flower to sort of move our research paper from the research paper to actually see if we can make a production level unit. And so this is what the Flower Federation looks like. And so again, this is Flower 1.9 ready. We're really super excited about seeing how we can improve on this. But effectively, what the federation looks like is you have your superlink, and your superlink is going to coordinate between your servers and your supernodes, which are clients, the servers that join. And be again linked to the servers already available. And you could start. You could say you have a server which is part of your research organization, and you're connected to different clients. Again, using SSL encryption, which is again provided by the Flower API for Flower 1.9, you can connect to the Superlink and start training and be a part of the federation. But say you want to now share your models or the tasks that you trained to a new research lab. And so all you would have to do is as simple as just sharing your certificate with the other institution, and they can connect as a new server. And when they do that, they will be able to download the best trained global model at the Superlink, and then if they want to have their own set of clients, they'll be able to add in and support training for new tasks as well. And all of this will happen while you already have a federation of supernodes and servers already training and training on different tasks. And so, this effectively shows that you can have clients add and join and even drop, and also completely work with heterogeneous data at scale. And so this is the demo that we presented at CVPR. This isn't a live demo. My apologies, but feel free to scan the QR code, and that will lead you to our GitHub. I'm just going to be talking about what are the steps and how easy it is to set this up yourself. And so again, all you have to do is just clone the repository, and you can have your environment set up. We've created a requirements file, so all you have to do is just install the requirements, and you should be good to go. And once you do have that, you can download the data sets. So in this case, we've shown the screen and the data set because they're pretty small, they're easy to download, and you can run this on a system that doesn't require too much compute. And once you have that, what you can do is you can run this experiment in a centralized setup, which is again talking about the centralized model that we trained, or you can run federated with the Fireworks API. And in this case, we actually show that we don't really have the SSH certificate case, but again, there's documentation to do that. We will be updating the GitHub repository to add instructions on that as well. But effectively, what you'll do is you'll start your Superlink, and once you start your Superlink, and you can have clients which are training for a particular organ, and you can start those clients and connect that to the Superlink and begin training. All right, so let's talk about the future scope. So we're really excited, how this has all come together, and we really would like to continue working on this. And so what we're going to do is we're going to add support for more models. So in the medical imaging segmentation community, a more common model to use, or a common AutoML technique to use, sort of AutoML for medical image segmentation, is NNUnet. And so we're looking at how we can sort of increase the scale and make this completely model agnostic, and also extend segues to clients with multiple modalities. So for parts of our research paper, we only chose CT images, but we also want to extend that to clients that will probably be bringing in magnetic resonance images or X-rays, and see how all of them can still be a part of the federation, and provide a global model that should be able to segment all of this together. One of the concerns that we noticed was standardizing the data. And so when you do have data heterogeneity, a big concern is how would you standardize the data that you're loading in, especially when you have such a variety of data. You have 2D and 3D data. And so that's also something that we're interested in doing. So the way that we're probably envisioning this is that we'll have a common data format. So irrespective of what modality and what data you have, you should be able to connect that effectively. And then we also want to investigate the performance of the global model as and when clients join and drop the federation, to ensure that you're actually training a global model which doesn't have problems with performance degradation as clients drop or join. And so, yeah, if you're interested in reading our paper, again, like Nick said, we just presented this at the DCAMI workshop at CVPR, where this was awarded the Best Poster Award. We're really excited, and if you do want to check it out, we highly recommend you read our paper, and you can get more information about how we've implemented this from a research perspective. And so, again, thank you to all the friendly folks who made this possible. I just want to have a special mention to Nick. Thank you so much for having us as part of the Flower Pilot Program. Special shout out to Javier, who's been super supportive and helpful in sort of building this infrastructure with us. And then to Vishwa Parikh, who is my PI, and who's the lead PI on this project. And also to every team member here at University of Maryland who helped this, with Dr. Yi and Sana Fulfani as well. So thanks to everyone who really made this possible. This is not a one-man show. And thank you for your time for tuning in and listening to this. If you have any questions or comments, I'd be more than happy to discuss.
Great, thank you so much. It was really, really interesting on, I think, multiple fronts. It was interesting on the front of research and also on the front of showing people how they can take some research idea and kind of make it a little bit more production-y and moving it forward towards actually deployment. Fantastic. Uh, just as we did with the other talk, if anyone in the room wants to ask a question, just raise your hand. I won't mute you, or you can use the Q&A facility here, or I'll look around to see if I can find other questions from folks in Slack and other places they can submit them. Let me see what I can find here. Oh, I see. Organ labeling. There's a question. I think it must refer to early on in your talk. They were asking a question: Is there a standard for labeling this organ data? Yeah, I said often this happens. People have different institutions labeling information differently. That can make the learning more problematic. So, in your domain, is that well established, or how did you deal with the problem if there was a problem?
Yeah, so we do have standardized formats for working with these images. So DICOM is the most common one used to use those, but there isn't necessarily a standard technique of doing this. This varies from clinician to clinician. So you could have some clinicians go above and beyond and literally segment every slice when you're going through it for these images, or you could just put them sort of segment a bunch of slices and then sort of extrapolate that to fit the entire organ. And you also have auto segmentation tools that some institutions will end up using to sort of help with the segmentation process. So it's really not standardized per se. There still is a standard format, but you can still see differences in labeling. And that is actually a big concern because, if you don't have, if like, as for any machine learning model, if you don't have clearly labeled data, that is going to affect your performance. And so we've tried to sort of develop tools that will allow clinicians to segment this in a better way. You have a lot of open source techniques, open source tools that a lot of institutions use. But again, this is not something that's completely standardized.
I see. Interesting. Cool. Cool. Um, yeah, I see there's another question about there was, I guess it's about organ variation. There was accuracy shown where there was some variation in accuracy with respect to organs. And the question is, do you know why that's the case? And see here the person put like, is it the size? Is it the number of pixels? Is it something else? Um,
Yeah. So I think I'll just share my screen again
oh,
to
yeah.
sort of read that. Yeah. So what we've noticed is that it definitely depends on the size of the organ. So if you can look at the table here, again, like I said, the pancreas is sort of a smaller organ, a lot more complex. When you're looking at these images, when you're looking at these images, you'll notice that when you're segmenting them, you'll notice how the view changes as you go by. For big organs like the liver or the kidneys or even the spleen, I would say the spleen is like a medium-sized organ. For bigger organs like the liver and kidneys, you see much better performance, and so that's why you see higher numbers in almost all cases. Whereas for the pancreas, you see that just because of the complex nature of how the organ is visible in the image, and also because of how small it is, you see not that great of performance.
Hmm. I see. Interesting. Yeah, because it's reasonably pronounced in some cases here, depending on
Yeah.
the
So
organ and the number of nodes and the technique being used. Hmm.
yeah. So I think just adding on to that is that we've noticed when you have multi-client setup. So for example, the 3-node and the 4-node setup. If you look at the 3-node setup, it actually is kind of counterproductive because the central aggregation technique completely fails to segment the liver and the pancreas together. but ends up somehow segmenting the spleen. And even for the 4-node setup, it ends up actually doing the exact opposite, when it doesn't segment the spleen at all. It just ends up probably segmenting the other organs. And so again, we feel that this is just because of how heterogeneous the data is. That bigger organs will be easier to segment, but when you're collectively training this and putting them together, that's really when it degrades the performance. And so just to add on to that, when you're training this, FL probably does a really good job of training this because of doing this in a decentralized manner, wherein you actually have, because you're doing this, the knowledge transfer at the server, it's kind of like the server is able to see different organs separately, different views, and able to aggregate that knowledge effectively, which probably central aggregation is not being able to do.
Right, right. I see. Interesting. Cool. Well, the one last question I actually had myself was that with your demo code that you showed, is it possible to can people use that code to stand up a very similar system, but then change out the task? Or is it more just a demonstration of what you showed us so far?
Oh no, definitely. So this code is again is completely is modular, and you can change it to have any task that you want. We designed it specifically for medical imaging, and so that's why you see this. And because of we wanted to test out the research that we did in our paper. That's why I showed today with the data set that we used for a research article. But you can use this. You can change the algorithm backend, so you could change Fed average, and you could try some other technique. And again, because this is supported completely with Flower 1.9, it's as simple as changing just a single line of the server to change the aggregation strategy. But you can also change the data sets. And so all you have to do is just probably for now, all you will have to do is just go into your client app and provide techniques to pre-process that data. So we're still working on that. So we're going to try to make it, again, completely agnostic. You would just have to feed in your data set and probably just give in particular variables, like, what is my intensity values for these images? And you have the script do this automatically. But for now, you just have to load in your data set and then give it certain pre-processing techniques that you would like to do. And then you'll be able to launch the client.
I see. I got you. Yeah, it makes sense. That was what I kind of had in mind. But yeah, okay, excellent. Oh, I was going to end things, but I see we have two other questions, so let's take them. One question is written down here. It says, "Great talk. I was just interested if there was a specific reason in choosing a multi-head unit encoder for the task. Given the multitask nature of the task, I was curious if something like an MOE will be better suited for this application, or was the intention to create a single, more lightweight expert for organ segmentation?"
That's a great question. Yeah, so I think the intention was to sort of make a very lightweight model, and so that's why we ended up choosing a mono unit at the back end, just because it's really modular, and we ended up choosing the multitask approach because of that. But yes, you could try different techniques. You could try a completely different backend, and that's something again we're interested in seeing. And so we're actively working on the next steps to see how this would work with a completely different backend. But again, we chose the UNet because that's just such a common technique in segmentation. That's usually the first default go-to technique that you would use. And so that's why we ended up using that. And also another concern was sharing these weights with the global model. We know that there's significant overhead when you're sharing this, when you're communicating with the central server. We wanted to reduce that, and that's also why we chose a simple unit in this case.
Got it. Sounds good. Let's take one more question. This is from Sanjeev. I'm going to allow him to talk, and he can ask his question for himself. That's great. So, Sanjeev, you're online.
Yeah. Hey, great talk. One quick question is, you mentioned selective weight aggregation. Can you just re-summarize that? And also, I mean, were these all weights in a one particular layer? How, are there some sort of known statistical techniques for identifying sort of the best selective subset of weights to aggregate, or whichever are affecting the gradient the most, or so on, right? So can you sort of, how generalizable is that if we wanted to do selective weight aggregation for all kinds of applications, possibly including generative AI applications? So just kind of more context on sort of the basis behind the selective weight aggregation.
Got it. Yeah, that's a great question. Thank you. Yeah, so for this study in particular, what we did was we empirically determined what would be the weights that would be important for being part of the representation block and those being part of the task block. And so based on our analysis, we realized that at least we wanted the UNet to have a good enough amount of encoder information while still being able to generalize and make personalized FL. And so that's why, again, this was all empirically determined. But yes, you do have smarter, intelligent strategies of how you can go about selectively aggregating the weights. What we usually see is you could also look at, say, running experiments when you would create a sort of sampling strategy, like to select which weights would be important, and then you would be able to run some sort of any sort of statistical technique to sort of verify. We haven't tried this, we haven't done this really, so but that's I think an avenue that we can definitely look into, and I think it's really important. You could also try something like a core set, for example, to determine what would be the effective size that of the layers that you would want to aggregate. And yeah, I think that's what comes to my mind. But I hope that answers your question.
Oh, great, great. Well, thanks for that final answer. We're running a little short on time, so I'm just going to move on. Thank you again for the great talk.